Hello there, I'm Chris Mancher, a master's student at the Department of Biological Sciences at the University of Manitoba, and I'd like to introduce you to RNA interference, or RNAi, a newly developing technology that is becoming a promising alternative to the fungicides used today. But before we delve into what is RNAi and what it means for Canada's pathogens, let's look into why we are researching this new crop protection technology. Producers across Canada are constantly being challenged by diseases brought on by many different pathogens. Fungi and oomycetes such as powdery mildew, downy mildew, sclerotinia, and blackleg are responsible for many crop losses if left unchecked. But fortunately, we have ways to control them and mitigate their spread. Fungicides are one of the most common and effective control methods, but they are not without their drawbacks. They could affect non-target species, or the pathogens themselves could acquire resistance. If these fungicides become ineffective or become banned, producers will be severely limited in their ability to protect their crops. So, what is RNAi and how does it solve these problems? Where chemical fungicides inhibit enzymes or disrupts metabolic pathways, RNAi operates by targeting the mRNA that creates those proteins in the first place. Once you introduce double-stranded RNA to the pathogen, its own processing machinery uses that dsRNA as a template to locate the complementary mRNA and degrade it, resulting in the silencing of that gene. There are many ways we can apply RNAi as a crop protection tool. Two common ways is to genetically modify plants to express these dsRNAs in their own tissues. This is known as host-induced gene silencing, or to topically apply them as a spray, known as spray-induced gene silencing. By using this next-generation technology, we can develop species-specific fungicides based on the unique sequences of each organism. As part of my research, I am investigating how we can use RNAi to control Hyaloperonospora arabidopsidis, the downy mildew of Arabidopsis. This obligate biotrophic pathogen begins as a germinating oospore from the soil, where it begins to colonize Arabidopsis intercellularly. Later, Haustoria begins to infect cells to gather nutrients, and then conidiophores produce asexual conidiospores that initiates new rounds of infection every 1 to 2 weeks. To protect Arabidopsis from downy mildew using RNAi, I selected 10 gene targets that have a role in either pathogenicity, cell wall formation, or are related to fungicidal modes of action. They were also selected based on RNA sequencing data from the reference genome that showed consistent or high expression in the first 5 days of infection. Using pipette tip boxes as containers, 15 2-week-old seedlings were treated with a 20 nanogram per microliter spray of dsRNA, inoculated with spores, sealed with micropore tape, and incubated for 7 days at 17 degrees Celsius. After, 3 seedlings were pooled together as one biological replicate, and RT-qPCR was ran on those samples. Of the 5 treatments processed so far, we have observed one demonstrating transcript knockdown compared to our GUS negative control, which is a dsRNA targeting the GUS gene and is not present in downy mildew or Arabidopsis. Relative expression is measured using actin as the housekeeping gene. To assess these treatments' impact on infection, we stained leaves with trypan blue and found that there was negligible differences compared to our control. What we suspect is that after 7 days, the expression levels of other genes begin to recover to normal levels or become overexpressed to compensate. Another suspicion we have is that there is not enough dsRNA delivered to the pathogen via a topical spray, as the dense trichome layer on small Arabidopsis seedlings may prevent dsRNA getting to the leaf surface. To address some of these problems, we began to investigate the growth of downy mildew at earlier stages. A previous paper displayed a way to examine spore germination on small cellophane pieces overlaid on MS media, and we became interested in doing something similar. We were able to replicate their protocol, but found that this type of imaging would be cumbersome to quantify the data, due to bacterial and plant contamination in the spore solution collected from plant tissues. This led to us using calcifluor white, which stains cellulose, a major component of the spore cell wall, and when using a DAPI filter set on a fluorescence microscope, we can clearly identify spore germination. What we ended up with is an in vitro method to accurately quantify spore germination and germ tube length by looking at 5 microliter droplets containing approximately 100 spores underneath the ImageXpress imaging system. We acquired multiple high-resolution images, stitched them together, and then scored them using ImageJ software. In our first test, 3 droplets per treatment were imaged at 48 hours post-inoculation. Interestingly, germination never peaked above 50% even in the GUS non-targeting negative control, and we saw multiple dsRNA treatments showing inhibition equal to or less than our fungicide positive control, metalaxyl. We can also see that germination tube length is also impacted by the treatments, and in situations where we did see some degree of germination, the germ tubes were much shorter. Our next steps are to run these tests again to see if we can obtain similar results. With this experimental design, we can examine many unique interactions regarding RNAi or other treatments and could be a basis for high-throughput functional studies investigating downy mildew growth. In other studies, we have seen the use of long dsRNAs to inhibit growth of plant pathogens. However, one of the many questions that needs to be answered is, how do these molecules get taken up by the fungus? Could pathogens alter uptake to develop resistance? And can we design different molecules to get in via a different way? For these questions, we're going to be using Sclerotinia sclerotiorum as a model, a destructive necrotrophic pathogen affecting canola. Let's take a look at 3 different scenarios regarding RNA uptake. For all of these tests, we grew Sclerotinia in broth culture for 3 days with its associated treatment and ran RT-qPCR on the samples with beta-tubulin as a reference gene. The target being measured is an aflatoxin biosynthesis gene, previously used in RNAi studies within our lab. In the first one, we introduced long dsRNA to Sclerotinia on its own and measured knockdown. From previous work by Nick Wittink, we have found that clathrin-mediated endocytosis is a primary mode of uptake of these long dsRNAs. The dsRNA becomes enveloped by a clathrin-coated vesicle and is released into the cytoplasm, where then it causes RNAi-mediated knockdown. In the next test, we treated Sclerotinia with a chemical inhibitor of clathrin-mediated endocytosis. Since we know clathrin plays a role, we expected to see reduced knockdown of the gene transcript we are targeting. We tried this with 2 different chemicals and found similar results, that the long dsRNAs are no longer getting in. For this third test, we did not use a long dsRNA, but instead used paperclip or hairpin RNAs containing a loop structure on either one or both ends. These dsRNAs consist of a 23 nucleotide region of the long dsRNA. When we treated the Sclerotinia with the clathrin inhibitors, we still saw knockdown, leading us to believe that these get in a different way, and do not use clathrin-mediated endocytosis as a primary mode of uptake. So, the question needs to be asked, by what mechanism do they get in? To answer this, we first decided to look at chemical inhibitors of other mechanisms of uptake. We treated Sclerotinia broth cultures using the same procedure as before, and assessed knockdown using either long dsRNA, short interfering RNA, hairpin RNA, or paperclip RNA. What we have found in the treatments processed so far is that no discernible pattern of uptake between long dsRNA and the short RNA molecules has been found, indicating that those mechanisms are not playing a major role in uptake. We also suspect that due to the toxic nature of some of these chemicals combined with the aflatoxin biosynthesis RNAi target, Sclerotinia growth may have been inhibited so much that we could not get enough material for qPCR. To solve this, we need a more specific and targeted approach. Instead of using chemical inhibitors, we can use RNAi to induce knockdown of uptake pathways, then measure knockdown of our target gene. This concept is called RNAi of RNAi, a 2-step RNAi-mediated experiment. First, we treat Sclerotinia with a long dsRNA targeting an uptake-related gene, allow time for transcript knockdown and subsequent change in protein expression, and then treat the Sclerotinia with the different RNA molecules that we then measure its expression. Based on literature regarding uptake pathways in fungi, I selected 6 genes to target with long dsRNA, each affecting either a different pathway or process related to endocytosis. To see if we can achieve sufficient knockdown with these dsRNAs first, I treated Sclerotinia broth cultures with the dsRNA and measured expression after 3 days. Each treatment consists of 4 biological replicates, expression is relative to a mock water negative control, and the reference gene used was UBC2, a ubiquitin conjugating enzyme. Of the 6 treatments, we achieved knockdown in 3 of them, and also saw expression of almost 200% in CDC42. We suspect that CDC42 is tightly regulated and plays an integral role in endocytosis, and I would be curious to see if its overexpression actually facilitates better uptake. Our next steps are to perform the RNAi of RNAi experiment using the dsRNAs that successfully induce knockdown, as well as take a look at the CDC42 target and measure any differences in expression of our long dsRNAs, siRNAs, hairpin and paperclip RNAs targeting our aflatoxin biosynthesis gene. Moving forward, we can then fluorescently label the RNA to observe its fate in uptake, and further explain the mechanisms involved in uptake within Sclerotinia. I'm excited to see where my research leads and what new questions may arise in regards to RNAi-based crop protection and uptake of dsRNAs, and look forward to sharing our results with you all. I'd like to thank Dr. Steve Whyard and Dr. Mark Belmonte for their mentorship and help in the conception of these experiments, the Whyard and Belmonte lab members for their contributions to my research, and to the Canola Council of Canada and the Natural Sciences and Engineering Research Council of Canada for their funding. Thank you.